Okay, so today we're going to have a data analysis tutorial for assigning cell types with a new tool called Garnet. This is going to be a purely Garnet tutorial, and we won't be covering any aspects of Monocle 3 in this, but we will have further tutorials on Monocle again. Um, yeah, let's get started. Okay. So we know that in the recent years, whole and multi-tissue single cell expression studies have become increasingly common. They have grown on a tremendous scale, and it has become imperative to come up with ways to accurately classify cells, especially in large complex organs like the brain. Although there are many tools out there, as we know, there is no set standard for using any single tool, and different people have different tools that they like to use. It's same as the case for cell type classification. I'm sure there are a bunch of tools out there, but today we're going to talk about Garnet. This is a tool that's been written by Hannah Pleiner in the Trapnell and Shinbury labs when she was doing her grad studies there. And hey. Okay, so this tool helps to cell type classify all kinds of single cell data, not just skRNA-seq, but I'm going to talk about the complex organs that skRNA-seq has been able to resolve cell types in. So the Cowpacker Science 2017 paper showed that skRNA-seq is used to resolve nearly all of the cell types in the worm, and this was a really big whole organism skRNA-seq experiment that was published around 3 years ago, and skRNA-seq also resolves neuron subtypes very well, and that's very important because just having a high level knowledge of what the cell type classification looks like in a very complex tissue is not enough. And you would want to classify it further into their subtypes and see the expression changes in your own data set. So if you want to do any kind of cell type annotation, the usual annotation workflow involves a lot of hard work. It involves a clustering of the cells. Then you have to find specific markers after all of the filtering that you do that we've spoken about in the Monocle 3 tutorial. Then you have to read innumerable papers about on Google about these specific genes that you find in the clusters. And then take your best guess for each cluster and what sort of marker genes are the best represented in each cluster. And that's still going to be a guess if you don't know a lot about the data set that you're working with. So what makes this a hard problem? One is that single cell data is sparse. So many specific marker genes that you'd like to find aren't at a whole animal level. And some of the highly expressed genes in many of the tissues or organs are highly leaky. So this data is from the Tabula Muris Consortium. And if you look at the albumin expression here, you can see that it's expressed in this cluster here. But when you look at whether this gene is expressed or not, it shows that it is expressed in other clusters as well, which shows that it's highly leaky. So this is where the Garnet tool comes into the picture and sort of helps you navigate the cell type classification without having to, or rather reducing your Googling. I wouldn't say you won't do any, but it helps substantially. So basically, Garnet uses a simple grammar for specifying cell types. what you use is a simple tab separated text file called a marker file. And uh, in the marker file, what you do is you specify the cell types, and you can also specify if there are certain marker genes that are expressed or not expressed. This is if you know a lot about your data. You can also specify the subtype for like if you in this case CD4 T cells, you can also specify that they are subtype of T cells, so that Garnet will later on hierarchically classify them. And I've not shown here, but you can also specify what tissue it came from, and the sample of origin, and then expression thresholds. So something like this gene is expressed above uh, this expression level and expressed below a certain mark. So if you have corresponding bulk RNA-seq data, that will help a lot. And I'll talk a little more about this when we go over the markdown. So basically, it's a very simple file to start with. It's a text file. You can make it in any text editor. So you don't need any complex tool or a very complex knowledge of R to do this. Then the Garnet marker file defines a hierarchy of cell types. So basically, this is an example of a marker file. So if you can see the different cell types are CD34 NK cells, monocytes, B cells, and T cells, and the subtypes of T cells. So what it does is there is a root node from which it says that these are the sort of child nodes. And then since you have mentioned in the marker file that the subtype of T cells is CD8 T cells and CD4 T cells, it'll classify that as sort of the grandchildren of the root node. And this is all happening in the background in the algorithm. So what Garnet does is it trains a multi-level classifier to subtype the cells. So what it does is first it finds from your entire data set, first it finds a representative cells as child nodes, which I talked about in the previous slide, that are these. So using the child node markers, it classifies the genes into the different types, B cells, T cells, and what this out group means is that it has mixed assignment, so it's not able to tell exactly if it's a B cell or a T cell. So it's going to call it unknown. And that's in the next step. So this is just the first step where the child node markers are used to just have an overarching classification. Then it trains a multinomial classifier using elastic net regression. So the intercept is your gene, and then these are your these are your cell types. And then using that, the classification of the cells is done at a particular threshold. So that classifies cells into the different types like B cells, T cells, and n number of cells. And then one set will be called unknown, where it can have like 10 types of cells in it. And then this is repeated if you have a very, very large data set, because you're going to have several clusters that are either very closely related or they are completely apart because they're completely different cell types. Then this classifier can be used on new data sets. So if you have one data set, suppose you have a 10x data set on version v1, and you train a classifier on that data set, and then you get the next one, let's say 1 year down the line, and you have it, you have run that on version v2, then you can always apply that classifier on the second data set without any issues. Yeah. All right. Oh, yeah. And one more thing to mention here is when it classifies the cells from the root node to the child nodes and then to the grandchildren, it uses a strict threshold from your expression studies to classify cells into their respective types. But another option that Garnet has is it extends annotations to cell clusters. So suppose you have a UMAP that has B cells, T cells in completely different clusters, and you have certain unknown cells in the B cell cluster and the T cell cluster. Then what it'll do is it'll extend that classification to the unknown cells, because it's very likely that these unknown cells are those respective types, B cells or T cells. So that is very helpful in large data sets where you have, suppose, like 20 clusters, which are very well separated and are clearly distinct cell types. That's when cluster extend option works very well to sort of engulf those unknown types, because it's just possible that the algorithm didn't capture them. So once you extend it, it's going to be considered as T cell. And you can always check it, because you can subset out the cluster and make sure that everything in there is a T cell marker, for instance. But in case of a very small data set where you have probably like one large blob of a cluster, and then you have B cells on one side and T cells on one side, and there's this small area where there are unknown cells, then it's not going to exactly know whether to classify it to B cells or T cells. So it's going to remain unknown. So you sort of have to take a call on when you want to use this option versus not use it.
Yeah, sure. I have a question.
Yeah.
Are there other ways that you can maybe change the thresholds to reduce some of the unknown cell types, or try and get them assigned to their correct cell type?
Yes. So if you have expression data either from single cell or bulk RNA-seq, then like I showed in the marker file, you can specify the expression thresholds and make it either lenient or strict, and have more cells be included in a particular cell type cluster.
Does anyone else have any questions at this point? I think this is a lot of information to get through.
Yeah. Oh yeah, and feel free to stop me anytime. Since our chat box is not working, you'll have to ask me questions directly.
Thanks, Ashwarya. So far, so good.
Okay. Okay, then I'll proceed. Okay. So like I said, Garnet classifies across platforms as well. So today we are going to use the PBMC dataset that comes comes within the Garnet package. So for instance, if you have trained your classifier on the 10x V1 PBMCs, you can use the same classifier to classify 10x V2 PBMCs, and it will give a very strong classification. Another property of Garnet is, or a feature of Garnet is, it can classify cross species and distinguish deceased versus normal cells. So if you see a classifier has been created here from a healthy mouse lung, and the same classifier can be used on a human deceased lung, and you can know what clusters in your data set are normal versus tumor. So this is a manual annotation of in the paper that was published, and the gray parts are the cancer cells. So when the mouse classifier was used on this data set, it classified all of the normal types properly, and then the gray ones are unknown, which should correlate with the cancer types. So you can subset out these clusters and look at the top markers from these clusters and their respective expression to know clearly that these are the cells related to the cancer model. Another very good feature about this tool is there is good documentation. There are already pre-trained classifiers on the website, and they also include the respective marker files for you to look at it has a GitHub link where you can post bugs if you've used the tool and come across something that doesn't feel right. And there's also a Google group where you can post questions. And this resource is continuously building. So if you have a classifier to donate, you can do that too to help build the resource. All right, so the first point doesn't apply anymore because we don't have a chat box. So just feel free to stop me anytime and ask questions. And I'll send a feedback survey after the tutorial, and we welcome any ideas to improve Zoom for you. But I guess I can hop over to the Markdown now, and we can start that because that's going to take longer than the slides. I'm going to stop sharing. Okay. Can you see the Markdown?
Yes, we can see it.
Okay. Cool. So the structure of this Markdown tutorial is going to be like main content, which I will talk about as I'm running through the steps of Garnet, the Garnet pipeline. There will be bonus questions, which you're free to attempt as you work through the tutorial with me, or you can do it later as well. And then there are challenge questions that are not very straightforward, and you need some additional information like some Googling and to attempt them. So you can go back and attempt and then email me or sign up for office hours to discuss. And I have some of the links that I think may be useful. So website links for Monocle 3 as well as Garnet. One of the things I'd like to mention is today we are using Garnet for Monocle 3. So the history of this is when Garnet was written, Monocle 3 was very, very new. So the first Garnet was written on the old Monocle, but then the old Monocle is almost out of service now. So everyone uses Monocle 3. So Garnet was upgraded to be compatible with Monocle 3. So I'd say use the docs for Garnet that are for Monocle 3, and that's a separate tab, and it's quite clear on the website. So these are website links for the two tools. And then if you have any bug reports, you can submit it on the GitHub repository. And you can ask technical questions on the two groups that are specific for Monocle 3 and Garnet. Okay. So I assume that everyone has all the installations ready. And so all the dependencies should already be installed. And the only thing that will be loading are the libraries for Monocle 3, Garnet, ggplot, and dplyr. Garnet, because we're using that, ggplot just to plot a few t-SNE plots later on. And dplyr is because some of the symbols that are used in the commands sometimes require dplyr to be loaded. All right, so I'm going to run through this now. Okay. All right, so an overview of the Garnet pipeline. So the Garnet pipeline has two major parts. It's not as complicated as the Monocle 3 because Monocle 3 does so many things that that it's not a straightforward pipeline. It has many offshoots, but Garnet is pretty straightforward, and it has two major parts. One is to train or obtain the classifier. So you either download an existing classifier or train your own. So to train a Garnet classifier, Garnet parses the marker file, which I mentioned is a very simple text file, and then chooses a set of training cells, and then trains a multinomial classifier to distinguish different cell types in the data set. And then the second part is actually classifying the cells using the marker that you've created in step 1. So Garnet applies the classifier to a set of cells to generate the cell type assignments. And then, like I said, there is a cluster extend option where Garnet will expand the classifications to similar cells and generate a separate set of cell type assignments. So part 1A is using a pre-trained classifier. The only thing you have to do here is read in the classifier. So if you already have one, then you can use that. But the whole purpose of today's tutorial is to learn how to build a classifier or train a classifier from scratch. So we'll move on to 1B. So there are instances when a classifier doesn't exist for your tissue type, or it doesn't include the cell types that you want in your data. So in those cases, you would want to generate your own classifier, and there are totally 5 steps involved in training your own classifier. So first is just loading your data. So it needs gene expression data from single cell data to get the expression of the genes from. So what we're going to do is we're going to use the play data set. Like I said, that's the PBMC data set that is within the Garnet package. So the system.file here is just because I'm reading the data from the package. But if you're using some other data or your own single cell data, you can just give the path to the matrix and the row data and the call data. Okay. So here what I'm doing is I'm just reading the expression matrix, the F data, which is the row data that has the gene information, and the P data that is the call data that has all of the attributes that you use for coloring your UMAP in the later steps. And this is the same function as seen in Monocle 3. So this just makes a new CDS object for you using the 3 files that we just loaded. So we are going to make a PBMC CDS using these 3 files. So if I look at the PBMC CDS, it has 32,700 genes and 800 cells. And it also tells you what the row data names are. So it has the gene short name in it and the number of cells expressed. And the column data has these columns right now. Okay. So the second step is actually constructing a marker file. So first major input is the single cell RNA-seq data. Second is the marker file. So it contains a list of cell type definitions written in a text file. And then these cell type definitions tell Garnet how to train the model. So each of these entries in the marker file starts with this caret symbol, and the cell type name is followed, and that follows the is followed by the cell type name. So it's basically this and then B cells. And then this is followed by like a bunch of things. So you can say whether this gene is, whether a particular marker gene is expressed or not expressed. You can mention the subtypes. So let's see how to build this marker file. So there are 2 major ways to build marker files. if you've attended the previous tutorials, and then you'll know that we get a marker file by using the top markers function of Monocle 3. So if you know a lot about your data, then you can just generate a file using known markers from the literature. And if you're super confident about it, then you can just go ahead. But if you're just doing sort of an exploratory analysis, then you can use the top marker function from Monocle. So run through the Monocle pipeline up to the top markers function and output the top markers result into like a CSV file. And then you can, or a text file, because that can be directly used into Garnet. And then the top marker function determines which genes are specific to each cluster. And you can just pick the top 5 to start with. So one of the common questions that we get is it better to have more markers for a particular cell type, like mentioned here, or fewer markers? So in my opinion, fewer very confident markers are always better than like 20 non-specific markers, because that's going to give very wonky results. So some of the things that you can add to your marker file are whether the gene is expressed or not, then what it is expressed above and expressed below. You can also have an entry that says it's expressed between two numbers, two specific values. More things that you can add is what subtype it is, and then what tissue it came from. Another thing that is very helpful to add when you're making your own marker file from literature is adding the references from where the marker file came from, so that you can always look back and remove or add any references and go back to the papers you got it from. Yeah, you can either mention URLs or like DOIs of the papers. Yeah, so basically the take home from this is a lot of things can be added in the marker file, and the more specific your marker file is, the better your classification is going to be. So I would say the revisiting happens in this step, where you make sure that you only proceed with a marker file you're almost 100% confident about.
Ashwarya, I have a question. This is Liz.
Mm-hmm.
Hey.
Hey.
For the values, what is it reading when Garnet takes those values? What in the CDS or what information is it using to determine those cutoffs?
So it doesn't have to be from the, okay, so it's going to take read in the expression values from the CDS, but those could be gene expression values from your bulk RNA-seq data as well. Does that make sense?
So I just want to make sure that if I'm putting in values, that it's correlating with the correct information from my CDS.
Right. So it's just expression values. So
okay.
You can say, so suppose in this case, expressed between. So this is 10 and 20. So it's possible that you're giving the low threshold for this gene as 10 and then the higher threshold as 20, because you have 3 different runs that say that it's expressed 8, 12, and 14. So you just want to give it a range.
Okay, that makes sense. Thanks.
Yeah.
I think this would also be useful for something like albumin that might be leaky and expressed kind of everywhere at a very low level. So you can just say, only call this whatever liver cell if it's above some value, right?
Right. So here, like you could give the albumin gene and then say that if it's above, suppose 5, only then consider it as a marker gene.
Yeah. And I would say generally when you're making these marker gene files, this is kind of the place where you kind of run it iteratively and maybe make a couple versions of your marker files so that you could say, like, if I add 3 more genes for each marker, does it make it worse or better? And right.
Yeah, and the more genes you add, and if they're not specific, it's just going to worsen your results. So I would say add more genes prudently. So this is the second part where you actually put things into your marker file and build it. And so now you have a text file that has all of the information you can provide it to have as confident of a result as possible. So the third step is actually checking and plotting your markers. So Garnet has a function to check whether the markers that you've selected in your marker file are likely to work or not. So the two relevant functions are check markers and then plot markers. So the check markers function, what it does is it generates a table of information, and then it has all the information about your markers, and then the plot marker function takes the most relevant information and makes a plot for you that tells you whether the gene is in the database or not, whether it's expressed or not expressed. So it basically parses all the information from your marker file and makes a diagnostic plot. Okay, so the arguments that are needed for using these functions are, one is database, so it depends on your model organism. So here we are going to use human, but like if you see this annotation database, so there are annotation database for several organisms, and if one doesn't exist for your model organism, you can always specify none, and it'll just take the gene entries from your CDS. That is why the expression data is so important, because it not only takes the expression values, it can also be used to take the ensemble gene IDs and gene symbols. So let's run this. So for using this database, I need to load the library. All right. So what I'm going to do is here, I'm just going to use a marker file that is already in the Garnet package. But again, if the marker file is created by you, you can just give the path to the marker file. So now I'm running the check markers function. So the inputs for this function are my CDS, my marker file, the database, and it's going to use the symbol for checking if the markers are good markers or not. And that's because if you use ID, it just doesn't make any sense, and we don't really know what gene it is. So if I do a head of the marker check result, this is the output that we get. So it has the marker gene. So I said use the symbol, so we actually know what the gene is. It'll also give its corresponding ensemble ID. It'll give the parent node. So do you see here, it says root when the cell type is T cells, because T cells is the child node and the root is the parent node. But in case of CD34, the cell type is T cells. And then you have something called nominates, total nominated, exclusion, dismisses, then ambiguates. So all this is scary to look at. So let's check what the marker check marker function actually outputs. So if you call the help function, then it will tell you what the check marker function does. So these are all the inputs that we provided. And okay, so this explains what the output data frame actually contains. So like I said, there is a marker file, there is the ensemble ID definition, then there is a parent column, which is the parent cell type in the hierarchical tree that we looked at in the slides. It also tells you the cell type the marker belongs to. One thing very important is to know if it's even in the CDS. So it tells you whether the marker is present in the CDS or not. And ideally, if you have not, so when you make your CDS and you've before making the CDS, when you've aligned your data, if you have not excluded any genes, it should ideally all be in your CDS. Then the nominates column. So these are the number of cells that the marker is estimated to nominate to the cell type. So that's a good thing. So if it's confident that a marker is good, then it's going to assign more or nominate more cells to that marker. Okay, then the inclusion ambiguous, it means that it tells you how many cells will become ambiguous if this marker is included. So basically high ambiguity score can mean that your marker is not specific enough, but there is a gray area here. So I'll talk about it when I come to that part. So, okay. So let's plot the markers first, and then I can talk more about it in detail. So plot markers, and then marker check. All right. So it gives you this sort of diagnostic plot. So if you can see, the genes are on the y-axis, and then the cell types are grouped here for all of these genes. And then there's something called a marker score and percentage assigned. So it's going to tell you if a particular gene is not in the database, or there is a high overlap with B cells when it's supposed to be T cells. So it's informative, because then you can go back to that gene, and then if by mistake you've put it in the B cell column, then you can just put it back in the T cell. So this is sort of helpful to either help you remove some genes or add other ones to replot this and check if that works better. So what are these ambiguity scores that we talked about? So what happens is when you check markers, so this check marker function, what it does is it takes your data, and then it picks a certain fraction of your cells as ambiguous. So it means that it may not have specific markers being expressed in them. So ideally, you would say that high ambiguity scores would mean that your marker is not specific, but that's not true. Sometimes high ambiguity scores can also mean that some other marker is the culprit. So you can check these ambiguity scores right here in the marker check output, and check what these particular cells are ambiguous with. And suppose there are two competing genes that have that have two different ambiguous scores, then you can check which of these gene is more specific for your data set and choose that to go into the marker file and remove the other one. So it goes through a lot of iterations before you can make a final marker file, is the take home from this. So basically ambiguity, the lower the better ideally, and the marker score is, it just tells you whether a particular marker is specific for your data. So the higher the marker score, the better marker it is. So there is no gray area about this. I hope that was not confusing. If you have questions, I can take a 2-minute pause here and take questions if there are any. So a couple of questions that come up in this section are, how can you check if a gene is a good marker or not? So these are all the steps that you should follow several times to know if a gene is a good marker or not. This is because your data sets may be very large, and repeating this step is way faster than repeating the subsequent steps. So I'd say some time spent here is better than a lot of time spent later. Yeah, and one thing you can do in this section is to know whether the gene is a good marker or not. You can use the plot cells function that we have used previously in Monocle 3, and sort of plot the expression of the genes and see if they are expressed everywhere or they're properly expressed in a specific cluster, which will give you the confidence that it's a good marker. Yeah, so you can either try this, or one of the things that you can check for is how can marker file building go wrong. So you can deliberately make, like when you're practicing, you can deliberately make a bad marker file and check how different it is from a very nice and clean marker file. So like I said, like what happens when you mix up markers. So if a gene is expressed everywhere, like the albumin one that I showed you, it's just going to skew your results, give you mixed assignments, and everything is going to look uh, gray or like unknown, because it cannot confidently put it into a particular cell type cluster. The results skew a lot even when you completely leave out a cell type. So if your data set has B cell and T cell population in large numbers, and if you completely leave out the T cell part, then it's still going to give you a very, very bad result. So be sure to include all markers properly. All right. So the fourth part, the fourth step to building your own classifier is to train the classifier. So the arguments are basically the same as the check marker function. Uh, yeah. So what I'm going to do is here is I'm going to set a seed. I did mention this in my previous tutorial. You set seeds to have reproducible results. So some of these algorithms in the back end use random number generator. So every time you run these steps, what happens is you can get slightly differing results, which can throw you off completely. It's just because it's not exactly picking the same exact cells every time. Uh, so what you can do is you can set a seed, and it can be any positive number. And every time you run that, it'll give you the same results then. So I'm going to use a final marker file that I call PBMC test as my marker file, and then make a PBMC classifier using the train cell classifier function of Garnet. So the input over here is my CDS, my marker file, the annotation database. Again, I'm going to ask it to use the symbol for denoting the marker gene column, and then run the train cell classifier function. So this is a very small data set. It only has 800 cells. So just imagine that when you have thousands of cells, it's going to take substantially longer to complete. Okay, so the training the classifier part is done. Now I'm going to go into the function and see what each of these input parameters mean. All right, so CDS, as you all know, it's your input CDS. Marker file is the file that you generated in the previous step. That's the text file. Database is the annotation database that has information on all the genes for your model organism. Then CDS gene ID type is the type of gene ID used in the CDS. So basically the default is ensemble, but if if an annotation database doesn't exist for your model organism, and if you have mentioned none, then this is just ignored, and whatever is in your CDS is used. All right. And then marker gene ID type is the ID type that you have written the markers in. So ideally it should be symbol because no one actually puts ensemble IDs in there. Now, num unknown, this is a very important parameter. It's an integer. So the number of unknown type cells to use as an out group, which is an unknown group during classification. So basically 50 is too small of a number and it's done only for speed, but the default is 500, which we should probably use because most of the data sets are quite large and not as small. So basically it's going to tell the algorithm how many cells to use as an unknown group. That's only to ensure that the rest of your cell type classification happens properly. And later you could always use the cluster extension option of Garnet to make sure that the unknown types or the cells in the unknown cluster are accounted for. Okay. So basically my PBMC classifier now has all of the information that I need to classify the cells. So one very nice thing to check is what does this num unknown parameter do? So if you change the numbers from 50 to 100 to 500, how does the result change? So to properly understand it, you'll have to actually do it to know the differences. And then you can see the output and see how the output varies. Okay. So after the classifier is ready, which is the PBMC classifier. So let's look at the PBMC classifier.
iPad, then. iPad.
Okay. All right. So the PBMC classifier is an object of class Garnet classifier, and it has all of the information that is derived from your CDS as well as your marker file. So the next step is the last step in training a classifier is viewing the classification genes. So like I said, Garnet classification is trained using a multinomial elastic net regression. So basically what this means in simple terms is that certain genes in your data set are chosen as relevant genes for distinguishing different cell types. So what genes the algorithm or the tool picks may be of interest. So because you want to be sure that you're moving ahead with the right trained classifier. So Garnet includes a function where you can see what genes are chosen as these relevant genes. So the function that is used is called get feature genes. So it basically takes your classifier as an input and says that start from the root node of the hierarchical tree and see if the feature genes collected at each point make sense. Okay, and then I'm going to do head of feature genes. So it's going to tell you an estimate, so an intercept of B cell, T cell, and unknown. So it's going to tell you if these genes are relevant or not. So the higher the value, the better it is. So if you see get feature genes, let's check the function. So basically, if you see this is a table of coefficients within the classifier. So it tells you whether a particular gene that has been chosen to classify that particular cell type is good or not. So like I said, higher the value, the better the gene is. And then one of the things that you can do when you're using an outside classifier that you haven't classified is get references. So as you can put references into your marker file, you can also check for references that have been put by someone else when they build their classifier and you are using it. So all you have to do is run this function called get classifier references, and the input is the PBMC classifier, and you'll have a list of publications from where the B cell and the T cell marker files were picked from. Right. So one of the questions that arises from this section is, there are certain classification genes that you have chosen which go into your marker file. So what running this function, get feature genes, this is where you can know what other genes other than the ones you picked were used to classify your cells. Because like I said, when you're checking for the markers in the previous step also, it's just quickly or like heuristically going to pick genes as marker genes. That's not the final number of genes that are used to define a particular cell type. So by using get feature genes, you can know, suppose it for T cells, it has picked 20 genes and only 15 of them were in your marker file. Then you can see the other 5 and sort of look at what the other 5 were that the algorithm picked for you. And that's very helpful to know, because it may be possible that you realize that those 5 that you didn't pick, but the algorithm picked for you, were also helpful. So next time when you build a new marker file, you'll remember to include those. Okay, so the part 2 is actually classifying your cells. So we're left with 2 parts now. So what I'm going to do is I'm going to take a quick 5-minute break, bio break, for anyone that needs rest. And we will gather back at, let's say, 10:05. And I'll go over classifying the cells and using a marker-free classification, which is a new feature of Karnet, and which may be helpful for a lot of projects. So yeah, let's take a break for 5 minutes.
Thank you so much.
Yep.
Thanks, Aishwarya.
Yeah, no problem. Okay, I'm going to start back up with the second half of the tutorial. So a recap of what we did. So we learned how to load the data, make your marker file, and then train your classifier in 5 steps, and the different checks to do at making your marker file as well as training your classifier. So all the steps that we did up to now should be the steps that can be repeated several times to make your analysis better, because when you actually do the classifying of the data set, classifying your cells, that's going to take a long time, and you don't want to have to do that a bunch of times. So the next step is to classify your cells. The function that Garnet has for doing this is called classify cells. So the input is your CDS, the PBMC classifier that you generated in the previous step. Again, the annotation database, and a cluster extend option. And I'll talk about that in a little more detail. And then again, what type of gene ID is in your CDS, and I'm saying use the symbol. So the cluster extend option is very helpful for especially large data sets. So what this option does is it tells Garnet to pick or sort of expand the classification to neighboring cells within the same cluster. So like I said, if you have 2 distinct clusters that have 2 distinct cell types, but within the cell type there is an unknown component, then if you use the cluster extend type, it will expand the classification to those unknown types and engulf it into the cell type for that cluster. And that's very helpful when you have 10 to 15 different types, which are clearly separated in your UMAP or t-SNE plot. But if there's just one blob and very sparse data, then it doesn't help as much. So let's classify the cells using the classifier that we built. Okay. And then let's do head of data of the PBMC CDS. So if you remember, when we looked at the head of the p-data of the CDS, when we first loaded the data, it only had t-SNE1, t-SNE2, size factor, and the fax type in there. But then after we classified the cells, now we have three more columns in the call data. That is Garnet cluster, cell type, and the cluster extend type. So this extra column is basically added because we use the cluster extend option true, and it won't be added if it's false. So it gives very confident cell type classification for large data sets. So it's like, I would say if you have a very large data set and you're not using the cluster extend type, you'd probably get 70% accurate results. But if you do use the cluster extend type, then you may get almost 95% accurate results. And this has been checked several times on large data sets. So let's do a table for the data of the PBMC CDS, and then let's say cell type. So it tells you how many cells are assigned to each of your cell types in your PBMC CDS dataset. So there are 200 assigned to B cells, and then the entire T cell population has about, I'd say, 200, and then there are 380 unknown. But now when I do a table for, let's say, cluster extend. So do you see the unknown factor goes away? That's because it realized that there were some unknown in the B cell cluster and there were some unknown in the T cell cluster. So it just engulfed those and denoted them as B or T cells. So it'll just give you the actual cell types and there won't be any unknowns. And you can always validate this. You don't have to completely trust the algorithm and say, oh, I don't have any unknowns anymore. This result, I mean, you can basically say that I don't have unknown anymore if you have done your due diligence and filtered your data, removed all doublets. So after doing all of that, you can believe that you ideally shouldn't have unknowns. But if you have not filtered your data, there are very low UMI read cells present, and you have not filtered for doublets, then it's possible that the unknowns are actually bad data. So do all the initial filtering properly, and then cluster extend option can actually mean what it's actually supposed to mean.
Sorry, can I just ask a quick question?
Mm-hmm.
Yeah, so what happened to the CD8 T cells in the extension data? So you lost that cell class, right?
Yes. Yes. So what it does is it gives priority to the child nodes and not the grandchild nodes. So if CD8 T cells had the least number of cells, so it's possible that those were not confident enough, or I would say those were not enough number of cells to have a separate classification when the cluster extend option engulfs the entire cluster. So it's going to probably be it's going to be conservative and say that instead of calling 48 cells as CD8 T cells, I'm just going to call them T cells.
So so in some analysis I've had, a cell type that has a really small amount of cells.
Mm-hmm.
Is it possible to, so if that's going to get engulfed because of the extension, is it possible to then use a combination of these two techniques to?
Yeah, definitely. There is no hard and fast rule to use one or the other. You can use it in combination as well. Like one more thing I forgot to mention is when you make the marker file, you can use, I'll mention that later, actually. I don't want to confuse the, yeah. But you're right. Sometimes you can just have data sets that don't have enough cells. So it's not compulsory to use this option, I would say. And one of the things that you can do is if you don't use the cluster extend type, then you can dig into this unknown cluster and see what the marker genes are and try to get rid of the unknown cells by actually labeling them. So the only problem with that is the problem with that is if it's a very large data set, it becomes quite tedious to do that. Yes, and if it's a small data set, it's still possible to dig into it and then, look at the marker genes and see if they are good markers or not. But engulfing is better when you have like 20,000 or 50,000 cells.
Great, thanks.
Hi, I just had a question. The numbers of unknown in the trained cell classifier, does it play a role in the results that we get in cluster extend type?
As in, like, is it going to be the exact number?
Yeah.
It should.
Because for example, you had given 50 as the number of unknown. I had changed it to 70, but I have got a different number of B cells and CD4 T cells and T cells.
Exactly. Right. So 50 is used just so because we're doing it quickly right now, and we have just 2 hours. But I would say 50 is a very low number, and you should probably use the default, which is 500. But so the number here, number unknown, when you use the cluster extend type, ideally it should match and segregate or use those number of cells and sort of assign them to the different types you have in your data. So this should equal to the increase in the number of this. Does that make sense?
Okay, so the if suppose the data set is very large, is 500 always enough, or we need to do a regression type of analysis to find which number of unknowns is perfect to use?
Yeah, that's a good question. So I would say start, if it's very large, start with 500, and then you can decide if that's giving you fairly believable results, and then decide if you want to increase that number or not. But it totally depends on how large it is. But I would say don't go below 500 for not like non-dummy data sets.
Thank you.
Will the cluster extend type always capture all of the unknowns and reclassify them? Or
Yes.
might there also be? Okay.
Yes. So in the next step, I'm going to show you how the unknowns become or get assigned the cell type of that cluster as in a plot, in a t-SNE plot. Okay, so let's plot this so that you can actually see what the difference is between the cell type column versus the cluster extend column. So I'm going to do a quick plot of the two t-SNE values and color by the cell type first, which I'm grabbing from the p-data of my PBMC CDS. Okay, so if you can see in this t-SNE plot, I have colored by the cell type column in my call data. So it gives me B cells, these two subtypes of T cells, just T cells, and then it says unknown. Now, when I use, let's say, cluster extend type. Yeah, so do you see what happens? In all of the unknown goes away, but since it's engulfing the entire T cell population here, it does not want to make any errors, and it wants to be on the conservative side. So it's going to get rid of the CD8 T cells because they were very few in number, and they're just going to have these cells, and the rest of the cells will be called T cells. So what you could do is subset out this T cell population later, and then sort of classify it into further subtypes. That's another way to go. Because in these type of cases, there are clearly two clusters. So it's, I can say with confidence that the unknowns are not actually unknowns, but they are part of my B cell or T cell populations. But then if I had this one blob of all of these together, then it would have been so confused that it does not know what to assign the unknown type to. So it's just going to leave it as unknown. So you'll have to make a call on whether to use the option or not. All right. So this is an example data set from a fact sorted sample. So we want to compare if these Garnet classifications align well with the true cell types. So there was a column in the CDS that's called fax type. So we're just going to color by fax type now and see if it matches. So this is supposed to be B cell, and this is the T cell population. And it matches. So this can also give you confidence that your cell type assignment worked. Yeah, so like we all discussed right now, pretty much this is the question that comes up in this section is how to know whether to use the cluster extend option or not. And I think the more you use the tool, the more you'll get to know its nuances, and the more comfortable you'll be in making a decision on whether to use the option or not. And when you plot the subsequent t-SNE plots like these, you can know how the difference or what the difference is, and if you can logically explain why you used one option over the other. And I would say it's not like one is better than the other, but there are certain advantages of using the extend type for large data sets, definitely. Okay, so the next part is an extension of the first part where we made our own classifier or trained our own classifier. This is a new feature of the Garnet tool where you can train a marker-free classifier. So this is particularly useful if all cell types are identified in one data set that you have, and then you want to apply it to a new data set. And you don't know a lot about your markers, and you have no information to give to your marker file, and that's fine. So you can just give a very, very broad cell type in your marker file, and it will still work for you. So let's see how to do this. So one point to note here is this is an alpha feature. So it's not yet published. So we encourage people to try it and get back to us on how it works and if there are any bugs. Okay. So some of the steps are going to be very similar to what I did in the part 1B when I was training the classifier. All right, so we have this CDS right now. So what we are going to do is we are going to make a new marker file. So basically, we don't know anything about the data. We don't know if it's expressed above or below a certain threshold or between two values. We don't know its subtypes. We don't know anything. So what I'm going to do is I'm going to add a new column called known type to my to the call data of my CDS. So it's going to be added here. And I'm going to say that from the fax type, if it's B or T cells, then add it to the known type. And you can add any type of cells in your known type. It doesn't have, since this data is fax sorted, I'm grabbing it from the fax type column, but you're free to add any other types. And then do a head of the CDS. So a new column called known type gets added. And then I already have a marker file that looks like this. So this is how a marker-free marker file looks like. I know it's very circular, but you still need a marker file. It's just that it doesn't have as much information as you would have if you're not using the marker-free version. So here, the only thing you're denoting is instead of the gene or expression information, you're just giving the known type as B cells. So ideally, we would give expressed CD4 or like not expressed CD8 or things like that. But when you don't know anything, then you just give that column in the call data and then say B cells and known type T cells. Another thing you can also do when you're using the marker-free version is you can have some cell type definitions in the marker-free version type and some as marker, let's say marker full version type. So you can have a combination of the two because it's possible that sometimes you have 10 cell types and you know a little bit about some cell type and you don't know a lot about the other. So when you have information that you can provide to help the process, then you can have the expression subtype not expressed details here. But if you don't know anything, then just give the column in your call data and say known type B cells or known type T cells. So this marker file is already generated, and I'm going to source that to the marker file path now. And then I'm going to make a new PBMC classifier using this marker file. Okay. And do you see the number of unknown changes? Because I chose unknown 50, and since I'm saying I don't know anything about my data, it's just going to consider it as 50. Because the only thing I said was the known type was either B cells or T cells. Right. So basically, this is the way you make the classifier with the marker free test. So the only thing that is changing is you specifying different type of information within the marker file. No other steps change, and the next step is just classifying your cells. So let's do that. So now it's just going to use the new PBMC classifier, which got rewritten when I ran the marker free version. And then, okay, let's try this. Yeah, so it's going to have a known type, and then based on the known type, it's again going to calculate a cell type and a cluster extent type. And let's plot it by known type now. So it still classifies it as classifies it correctly as B cells and T cells. So this is again, so when you use a marker free version, you would want to give the known type as B cells, T cells, and then you can add a bunch of entries like known type CD4 T cells or CD8 T cells, and those will get colored. But if you're just guessing and you don't know anything, then I would say have the child nodes as the only classification and you'll get the right classification. But you have to decide whether you want to spend a lot of time knowing what marker should go in, or just do this and see if it's actually working first. Okay. So that ends both the sections on training your classifier and classifying your cells. So one of the things that I mentioned when I was going through the steps was if your model organism doesn't have an annotation database, you can always specify none and it'll still work because it'll use the annotations that are present in your CDS. You can submit a classifier and it will be validated, and if it passes all the checks, then we can use it to enrich the resource. So feel free to submit classifiers, and the process to submit the classifiers is on the Garnet website. Another note is there were two papers that human fetal papers that came out using skRNA-seq and skAttack-seq from the Shendure and Shrapnel labs. So very soon, Hannah will be adding the fetal classifiers also to the website for you to use, and it'll include the marker files as well. I just wanted to cover one last part, and I'll have to switch to the PowerPoint for that. I just wanted to show you the sort of common marker file errors that you can spot in time so that you don't go through the process and have to redo it. So I'm just going to switch over to the PPT. Okay, so this is on the example data set was trained on the 10x PBMC version 2 data, and then it was used to classify the version 1 data. And so these figures or these UMaps are for are on the PBMC version 1 data of 10x. So all of the different marker file errors that happen are listed here. These are the most common. I haven't come across any other ones, but let's see. So the first panel is basically the fact-based 10x cell type assignments, which is the main data set that we're using, or the manually annotated data set. And the remaining panels, that's these 4, are colored by the Garnet cluster agnostic cell type assignment. So how do you spot when there is a clear mistake in your marker file and hence your classification? So one is if the cell type is not defined in the marker file. So the data that we were working on right now, we had 2 main cell types, B and T cells. So if you don't include the T cell definition at all, for instance, then it's going to give you this whole patch of gray here, which means that you've clearly missed out a very crucial cell type. So you'd want to go back and check what that is and include it. Second is if the cell type is not if the cell type is defined, but it doesn't include good markers. So if you, for example, define T cells using only CD4 and not CD3. So it's not that it's wrong, it's just that it's incomplete. So it's still not going to give you a very pretty classification. So you'd want to, if you can, you would want to give it as much information as possible and enrich your marker file to have a better classification. And then the third type of error is the cell type definition or the marker file doesn't include includes a non-specific gene. So for example, if you add a very widely expressed gene like MALAT1 or gene that is leaky like albumin to any one of your cell definitions, then what happens is you get mixed assignments. So it's going to be an unknown, either going to be an unknown group, which is completely grayed out, or it's going to be a group that has all of the cell types in it. So it's like a very colorful cluster, but it means nothing. So you'd want to go back and remove the widely expressed gene. You want genes that are specifically expressed in particular clusters to be confident that they're good markers. The last type of error is if you just by mistake switch some of the marker genes between B and T cells, for instance. This is the case where the definition is just wrong, but it's also a case which can be corrected very easily. So it's just like you included some genes for T cells in B cells, so it can just be rectified very fast. So this is a very wrong marker file, but a very easy error to fix. So these are the common 4 errors that we have seen people come across when they're making marker files. So be vigilant when you build a marker file, and go through several iterations. It's okay if it takes time. I think it's worth it in the long run, because that file is going to stay with you forever for that particular data set. And it can also be applied to another data set of the same tissue. So yeah, watch out, and good luck with marker file building. That's the end of my tutorial. I'll take questions now if there are some parts that I should talk more about. And I'm going to stop sharing.
Thanks, Aishwarya.
Yeah, no problem.
I have a feeling that I kind of know the answer, but how translatable do you think this is to data from single cell ATAC-seq?
So I would say it's going to help if you have both the attack and the RNA in parallel, because you do need expression data from the RNA-seq, either bulk or single cell. But I would say to make the CDS, you definitely need single cell RNA. So I would say if both the both data sets are present, it can be used to a good level of success. But with just ATAC-seq data, it becomes really hard for us to annotate all your clusters. It's a much longer process, I'd say.
Okay, thank you.
Yep. Okay, so I guess if there are no other questions, we can end the tutorial here. Feel free to contact me via email or sign up for office hours if you have questions. And if you attempt some of the bonus and challenge questions and are stuck somewhere, reach out and I'll be happy to help.
Thank you so much. This was really great. I really appreciate your time. Thank you.
Yeah, no problem.
Aishwarya.
Thanks.
Thank you very much. That was amazing.
Thank you.
Thank you so much. Have a great day.
You too. Thanks.